It's my great pleasure to come today and share with you our recent study on this how to use FFPE epigenomic profiling technology to study different human cancer and how to use it to contribute to, like, say, prediction clinical outcome so that we can use this technology and use the data in precision oncology. Okay, so you probably have heard of FFPE before. This is like the one of the should be the most commonly used biobanking technology to store the tissue. So, for example, in the operation room, after the doctor take out the tissue or biopsy from the patient, usually they are put into formalin and embedded in paraffin so that it can be stored for years or decades just in the room temperature, just like showing here. So you can just be put into a box and put it on a shelf and store in the room forever. So usually those FFPE tissue looks like either on the slides, put on the slide, you can see like in the middle, there's some tissue. Those should be that biopsy or some tissue and covered by the paraffin. Or it can be scripted up and stay in this curl format. So on one hand, this is such an easy way to store it in room temperature. But on the other hand, it is known for the degradation because during all those procedures, like in formalin procedure, the formaldehyde will degrade the molecule within the tissue. Paraffin is organic solution. It will also continue to degrade the tissue. And also over the years, the humid temperature will also like make the tissue become bad. So with that, like previously, there are usually two commonly used assay to profile FFPE tissue, either whole genome sequencing or RNA-seq. However, both of them are very affected by the degradation in FFPE. In other words, the DNA RNA molecule will be chopped up within the cell. So the reason we want to propose this FFPE CATAC because this is a DNA assay. So even the DNA can also be chopped up into small fragments. This technology can zoom in those fragmented region and then capture all those chromatin open region, or you can use an antibody so that you can capture, say, RNA-PO2 region. And because it can zoom in those very small region, give you a relatively small fragment, like around 120 base pairs long, it's less affected by the damage, by the degradation of FFPE procedure. And because we can use an antibody to directly capture the RNA-PO2 signal on the DNA, this is kind of like a direct measurement of gene transcription. Compared to RNA-seq, it uses those RNA molecules accumulated in the cell as a proxy of gene activity. Here, we can directly measure the gene transcription. And again, this is a DNA assay, so it will not be affected by those like RNA instability or RNA the transcript half-lives. So the data is relatively stable, and also data quality tend to be more like much higher than RNA-seq. So this is what the data looks like. Showing here is a genome browser track. So x-axis is a genome coordinate. So you can see like if there's a peak region, it represents there are like a relatively strong either histone or RNA-PO2 signal around this region. And for the rest of the region, you can see they're very minimum background compared to like say ChIP-seq or other assay developed for FFPE. So in other words, the signal to noise ratio for this assay is very high, very clean. So all those signals are very concentrated. And now we can like using the genome browser to broadly see the quality of the data, and we confirm this is a good data for the downstream analysis. Okay, so now we have the technology. We want to see like, can we use it to profile different like human cancer? So we take 7 different human cancer and have its adjacent normal tissue for comparison. And firstly, after we get the data, we want to see like, is it high quality data? Can it separate out different cancer type? And you can see like showing here is a UMAP. So each sample represents a cancer type. Red represent tumor, blue represent normal. So clearly like it can separate out tumor versus normal, and also it can separate out like different cancer type very easily. And also we check like the data is free from all those biases like library size biases or coverage biases. So it looks like a good data to continue. And again, like you can always zoom in to the region of your interest. So for example, we can easily observe there are hypertranscription among the HER2 region for the breast cancer. And similarly, we also see like some elevation, some hypertranscription of HER2 for colon, but we did not observe it for all the other cancer type. Speaking of the hypertranscription, again, this is genome browser track, red showing tumor, blue showing the adjacent normal. So we constantly found there is a region that tend to have hypertranscription in tumor versus normal, regardless whether it is a mouse brain tumor or different like human tumor. And those regions turned out to be histone genes region. So you can see like among those histone genes, it always have a high signal, like more peaky, like more enriched RNAPII signal in tumor versus normal. So that motivated us to dive into those regions and see like why histone genes always have share this hypertranscription feature in tumor, regardless the cancer type, and also regardless the species. So that's why we continue and focus on this histone gene region. So our hypothesis that the reason why we always observe hypertranscription in cancer versus normal is that when a cell that wants to replicate it, it does not only need to generate duplicate the DNA. It also needs to generate enough histone so that the DNA can wrap around the histone to stabilize the structure. Otherwise, the cell will die because the naked DNA is such a fragile structure. And for us human, we only have like 64 histone genes. However, all those histone genes encode all 5 histone subunits. So that need to be generated in such a efficient way so that it can match up with the duplication of the DNA, and then so the DNA can wrap around it to form a nucleosome so the cell can be replicated. In other words, regardless whether it's a cancer cell or normal cell, we need histone to pack the DNA. So for a cancer cell, if it wants to proliferate very fast, it needs to generate a histone in a very efficient and fast manner. So that's why we kind of hypothesize like histone production can be the rate limiting for the S-phase progression. So in other words, the reason why we always observe hypertranscription in tumor versus normal because the tumor cell needs to generate enough histone to keep up with the requirement for packaging new DNA in cancer cell so that it can proliferate faster than the normal cell. So that's our hypothesis. That is totally motivated by what we observe on the genome browser data. So to start this hypothesis, because we kind of feel like if the tumor cell want to proliferate fast, it needs to generate histone in a fast manner. In other words, histone genes need to have hypertranscription. So in other words, can histone genes be an indicator for the tumor aggressiveness? High signal of RNAPII at histone genes represent fastly progressive tumor cells. Can we use that as an indicator of tumor aggressiveness? So to start this hypothesis, we use a relatively simpler cancer system called meningioma. Not only because meningioma is one of the most common brain tumor, and also most people having meningioma, their tumor are mostly benign, but some of them still come back and recurrent, and those tumors become quite aggressive and malignant. However, the histologic data just shows like those tumors just look like a benign tumor, but they can still come back, recur, and become very severe malignant tumor. So that's why, besides histologic data, can we provide more like orthogonal genomic information so that we can, for example, based on the histone genes, we can provide orthogonal information to predict the recurrence. Again, like after we get the data, we want to check the data quality. And showing here, like those red flag represent the meningioma sample. And we have like 30 patients. In total, we have 36 samples for meningioma. And showing here are 2 units to start in the meningioma. Left one is like we bin the genome. This is a whole genome, so we bin the genome by 500 base pairs. And you can see the different cancer type can already be separated. However, if we zoom in more meaningful region using the cis-regulatory element region, this is defined by the encode. So you can see we can have a much better, not much better, like improved separation of meningioma and all kinds of other different cancer type compared to the normal. However, if we zoom in, only look at those histone genes, the 64 histone genes. So all those like separation of different cancer type just went away. Instead, we see like the separation between normal and tumor. That represent like the histone genes information probably is shared across different cancer type. So those kind of information motivate us to dive into the histone genes further. And also you can notice that there are some tumor sample that kind of like migrate into this tumor, the normal cluster. So that makes us curious whether those tumor cells are more like a benign tumors. And for those tumors that are very far away from the normal cluster, are those tumors tend to be more like malignant, like more aggressive tumors. So with those questions, we continue to study the histone genes. So because to study whether the histone genes, the signal, RNA-PO2 signal as a histone genes, whether they can predict clinical outcome, we definitely need an outcome to tell us whether there is a prediction power. However, like for those 30 meningioma patients we collected in Seattle, those patients are relatively new. So none of them have recurred. You need to wait like 5 or 10 years to see any recurrence information. So in other words, our data have very minimal clinical data. However, because this meningioma is such a common brain tumor, their literature study has been gone through so many years. So we just curated all those public available RNA-seq data that do have clinical information, including the recurrent time and status. So in total, we collected around 13 public data sets. And in total, we have around 1,300 public RNA-seq data for 1,300 patients. And those are mostly RNA-seq data on the frozen sample. In other words, the data is relatively of high quality. And some are FFPE, we check the data quality is relatively low, so we mostly exclude them from the integration. So now the task is that we have our data that can profile on the genome-wide manner. And we also have the public data that come with the clinical information. How can we harmonize those two modalities so that we can integrate our data, our epigenomic data, with the clinical information? So the integration is never a stranger for a computational analyst. So to integrate our FFPE tags, those epigenomic data with RNA-seq data, we just use those canonical correlation analysis, those kind of methods that are originally developed, say, by Surratt to integrate single-cell RNA-seq data with another single-cell RNA-seq data. We just simply applied here, and it turned out the integration outcome is quite ideal. So, for example, showing here is UMAP. And if a patient start with C, if a sample start with C, it represents it's a patient C. If the sample end with CATAC, meaning this is our technology FFPE CATAC, the epigenomic profiling. If it end with frozen, meaning that this is RNA-seq data on the frozen sample. So you can see regardless what kind of assay you use to profile the data, they can all be very nicely integrated as long as they are from the same patient. So this is for patient C, patient Z, patient I, X, P, J, B. So you can see not only they can be integrated very well, they are almost at the overlapping pattern. So in other words, we can very nicely integrate FFPE CATAC and RNA-seq data so that we can bring in the clinical information to harmonize those data together so that we can use the histone genes to predict the clinical outcome in the downstream analysis. Okay. So now we have harmonized those two modality data. The next is can we predict the clinical outcome using the histone gene signal? So before we do that, we look at the genome browser across all kinds of histone genes in a more careful manner. So showing here, are histone genes on chromosome 1, and also there is another histone gene cluster on histone 6. So you can see like this is ordered by the histone genes, overall histone genes signal, and colored by the WHO grid. In other words, like when a patient comes to a doctor will give it a score based on WHO grid criteria. So the more redness, meaning that the more severe the doctor diagnose this patient to be. So you can see like relatively have this like correlation, like if the signal is higher, it tend to be more severe. If it's more blue, meaning that it's more benign, and the signal become like more minimal. So in other words, we kind of feel like that there's no very dramatic like variation from histone gene to histone gene. They all follow this relatively decreasing pattern as WHO grade the severeness drops. So I just like use a very simple summary statistics that takes a median of all those signal across 64 histone genes. So with that, we can come down to one histone gene signal score. And using that score, histone gene score, and generated this density distribution across all 36 samples of the patient. So in other words, x-axis is summarized histone gene score, and if it has higher density, meaning that there are more patients that tend to have this kind of score. So now the next task is that how can I set a threshold so that about this threshold, I just predict all the patient to have aggressive tumor. And I just like come back and look at the meningioma feature, and it tells me like most of the patients should be benign. So in other words, if I want to set the threshold on this density distribution, it's better to set a threshold at the right-hand side of the peak so that most of the patients are still be predicted to be in the benign group. So I just randomly set it at 4.4. So the extra threshold does not matter. The order matters more, which I will elaborate like more in detail later. But now I just randomly set the threshold at the right-hand side of the peak and predicted all those patients on the right-hand side to have aggressive tumor and all the rest of patients on the left-hand side to be in the benign group. And let's see what does the clinical outcome tell us. So again, the UMAP shows us the integration of FFPE-CADAC, our epigenomic data, with RNA-seq data. So the circle are FFPE-CADAC, the triangle are RNA-seq data. So based on this very random arbitrary thresholding, around 5 patients are predicted to have aggressive tumor, and they are highlighted here in red, a red circle. And then I find it's a top 20 years neighbor in the RNA-seq data, and also predict those 20 years neighbor of aggressive patient to be aggressive as well. So all those red dots and a red triangle are predicted to be aggressiveness. And then I want to compare between the aggressiveness and the benign group, including all those FFPE and the RNA-seq data. So here is the survival, the recurrence information. So using the KM curve, you can see like for those patients that have been predicted to have aggressive tumor, they tend to recur much faster than the benign group. And also, it kind of like somehow matches even the Wikipedia description, like 5 patients fall within the 10 to 15% the population level of meningioma tumor being like more malignant instead of more benign. So yeah, you probably want to ask like the way you set the threshold is such a arbitrary and data-driven manner. What if I want to set a more stringent or more relaxing threshold? What will happen? So the consequence is that like how to set the threshold, whether we need how to give a threshold that have like clinical indication that need like more sample. Imagine like now we only have like 30 meningioma sample. It's very hard to to propose a like universal threshold that doctor can use in clinic. But what we think that matters is that the order of the sample that is ranked by the histone gene signal, the order matters more. So in other words, if the patient have more histone gene signal, it tends to be associated with more aggressive tumor, or like a patient with relatively lower histone genes, they tend to be associated with more like a benign tumor. That order should matters more than just propose like a universally applicable threshold. So in other words, if you try using different threshold as I indicated here using those dashed line, you will see like using more stringent threshold, you will have much more separation power between the aggressive group and the benign group. As you relax the threshold, you will see like the separation power just went away. So there is no separation between the benign versus tumor. In other words, you can see this kind of order of histone gene signal that really represents the aggressiveness of the tumor. So in other words, the order of the histone gene signal matters more. And also we have other like negative or positive control exploration. For example, you probably will be curious, like there are so many other oncology related genes or tumor aggressiveness genes, what happens to them? So for example, we look at the ribosomal protein genes and using similarly thresholding matter and using like the top 5 patients and predict them to be aggressiveness, and we did not see a separation power. And again, there is such a like a theory like Warburg theory, saying that a tumor cell tend to consume less oxygen. So in other words, we use a percentage of mitochondria, and again, we did not see a separation power. And we do have a positive control because around 70% of meningioma patients, they will have this chromosome 22q chromosome arm loss. So most of the patients who have this loss tend to be tend to have like quite severe meningioma tumor, and we do see like significant separation between patients who do have this chromosome 22q arm loss versus who do not have the chromosome arm loss. So this is our positive control. But the separation power cannot be compared with the histone gene separation power. Okay, so we have more other exploration on histone genes and association with aneuploidy. So all those detail, you can find it in our recent published science paper. It's already out, so you can just Google it to find more detail of the story. So to go forward, like right now, I have taken with me this technology to MD Anderson Cancer Center. So I'm running a hybrid lab. My wild lab focuses on this technology. So hopefully, because right now we have this hypothesis tested on such a naive tumor system called meningioma, but how well it can be similarly applicable to other tumor types, especially like more invasive tumor type like breast tumor or other solid tumor, still need further exploration. And those more invasive tumor tend to be more complicated because the tumor purity or tumor density will be very heterogeneous from patient to patient. And that is what we are exploring now. And at the same time, we're also setting up a matching computational pipeline to handle this particular epigenomic data. The major motivation is that this is tumor cells, so a lot of normalization or data analysis hypothesis assumptions will not be like very refined hold in tumor system. So that's why we're also trying to set up a matching computational pipeline. With that, we want to set up this FFPE epigenomic ecosystem to better benefit the precision oncology. And with that, yeah, our lab is definitely very open to all kinds of collaboration. Okay, so I will end with acknowledgement to my postdoc group, like Steve Hanikoff. You will notice that he is the first co-author on the paper. So he's the one who created this technology and really driving through this paper, this project. And also acknowledgement to my grant who's that supported, and also my about to establish lab at MD Anderson. And you can find my lab website here. It describes all kinds of projects. And we are actively hiring. So We are hiring for intern, postdoc, and staff scientists, and we have an application link available as well. So feel free to explore our lab website. And I will stop here to take any question. Yeah.